everybody. Hello and welcome to the first Getting to Know You, your scientific community webinar. My name is Kirsty Bourne and I will be your host. Today's webinar is sponsored by my company, Elim Bio. Elim Bio is busy supporting researchers on the front lines. We're proud to say that we are helping scientists solve the problem of COVID-19. During this unusual time, we also wanted to reach out and help feed future scientific curiosity. We felt the best way to do this for our at-home students is to do some virtual webinars with some cool scientists that are in our community. Today's panelist is Dr. Rafael Rojas. Dr. Rojas has his PhD in pharmacology from the University of North Carolina at Chapel Hill. His current research focus is drug discovery and precision medicine. And I was very lucky that before COVID-19, I got to visit Dr. Rojas at his lab. I've also been in the classroom with Dr. Rojas when he volunteered to teach labs like frog dissection. He's a very talented scientist, a great dad, and an all-around amazing human. Before I turn the mic over to Dr. Rojas, I wanted to clear up a little housekeeping about how Zoom webinar will work. On the bottom, if you scroll your mouse, there's a Q&A section on the very bottom. You have to scroll your mouse down to find it. And when you click the Q&A, you can type in questions. You can even type them while Dr. Rojas is talking, and I'm going to read them, and it won't disrupt him. And I'll read them during our question and answer period. We'll have a couple of them during the talk. And some of them I might answer on my own, but most of them I'll go ahead and feed over to Dr. Rojas. And you can go ahead and type questions that way anytime you think of them. And then I'm also going to throw up some polls every once in a while just to kind of get your ideas of things that he's talking about and maybe just some fun stuff about Dr. Rojas. So I'm going to go ahead and pass on over to Dr. Rojas so he can tell you more about what he's been doing and about his research. All right. Dr. Rojas, go ahead and take it.
Thank you, Mrs. Bourne. And thanks, Elim Bio, for sponsoring this webinar. It's really cool that kids that are stuck at home are able still to learn about science and get to see what people like me do. So we appreciate both of you guys taking the effort to do this. It's a really cool thing. So I'm gonna try sharing my screen. I have a couple slides that will go over what I do. And I think you guys might find a little bit interesting. So what I want to talk a little bit about is drug discovery, especially precision medicine. So what do you think of when you think drug discovery? Drugs are medicines. So these are not bad things. These are drugs or medicines that people take to make you feel better. And I've devoted my career to try to find drugs to help people with diseases, with problems, to help them. So currently, what I'm focusing on is what's called inflammatory bowel disease. So here's a picture from the website of the company I'm currently working for. Their name is Prometheus Biosciences. And what we do is we help people that have gastrointestinal problems, really severe problems with their GI tract. And these patients, they have to have surgeries. It affects people of all ages. In fact, we know someone that's very young that has what's called Crohn's disease, and it's a debilitating disease. And unfortunately, there's not a lot of drugs out there for these people. They don't work very well. And so what my company and what we're devoted to doing is find better medicines for these people. And we have a unique spin on this. We use what's called precision medicine. So what does that mean? There's a difference between precision and accuracy, right? What we're doing is we're trying to identify the patients that drugs are going to work for. So if I go to this next slide, one thing that is important when you think about drugs or anything is that one size does not fit all, right? I mean, the kid in the corner, that's not going to fit in. There's different size shoes, etc. So it's the same thing with drugs. If there's a patient that has IBD and they take a drug, it may not be the right medicine for that patient, or it might be a better medicine for somebody else. And so what we're trying to do is understand the genetics of someone so that we can design a better medicine. So we use the genetics of people. Interestingly, If you look at a human versus, say, a chimp, here's a human, here's a chimp, and you look at their DNA, it's 98% identical. I mean, that's very, very similar, but apes and humans are quite different if you look at them. But there's certain regions in the genome that are changed so that we look very different. And if you look at, say, one human versus another, their genetic makeup is even similar, 99.9%. Yet you look at me and you look at Mrs. Bourne, we look pretty different. So what's making people unique? If you go to the next slide, it's very small differences in our genetics that make us unique. So this is a slide showing 3 different people. This is a cartoon of their DNA. Remember, DNA is made of 4 bases, G, C, A, and T. And at this position, this guy has an A, this guy has a G, and this guy has a T. It's very small differences like that that make us unique. These small differences are called SNPs, or single nucleotide polymorphisms. So we identify different SNPs in people. We use genomics. We actually use artificial intelligence and bioinformatics to understand the genetics of people and try to find special drugs for those people. Now, an important part of what I do is drug discovery. So drug discovery is a process that takes a long time, takes up to 10 years, $1 billion, which is why medicines are so expensive. And this just outlines at a high level, going from left to right. You start with an idea, we want to help people with Crohn's disease and IBD, and at the very end, after all that time and money, we finally have a drug. The part that I'm most involved in is this early phase drug discovery. And I think this is actually the funnest stage, because at this point you're thinking, oh, I got this great idea for a drug. Let's see if we can make it work. Let's see if we can actually go through this process and identify something that works. And so this is what I work on right now: target selection, identifying compounds, and doing what's called lead optimization. So you have something that you think is going to work, you test it, get data, then you analyze that data. A chemist will make some changes, and you produce more data. It's a circle. It's an iterative circle. A lot of people involved in this. And so the cool thing about science is really it's a team sport, and we work together to do this sort of thing. So that's an overview of sort of what I do. I've always been interested in drug discovery or science in general, but what the spin that I like to do on it is I like science to try to help people. So, you know, if I was sitting around studying, say, you know, something just for the fun of studying, to me, that wasn't engaging enough. I wanted to have my research actually benefit people, which is why I went into drug discovery. So I work at a company. I worked at several companies. Before the companies, I worked in academic research labs, which are also fun and exciting to do that. But I really enjoy working in biotech companies. Yeah, and that's basically what we do for what we do. Do you have any questions?
Sure. So I had some of the kids said that we put up a poll, is accuracy and precision the same thing, which is kind of more of a math thing, but that was cool that, you know, a lot of them said no, that it's not the same thing.
That's good. They actually are not the same thing. And the way I like to think of it is precision is having all of your you know, if you think of like a dartboard and you're throwing darts, precision means that at least the darts are near each other, right? Mm-hmm. Accuracy is, you're close to the target. They're not on the target, but at least they're close to the target. So precision is all the darts are together, but they may or may not be where you want them to be. Right. But accuracy is, okay, now we're actually getting stuff there. So what we strive for in science is both precision and accuracy. We want to have all of our answers and our data be in the center and not over here. So we want to both precise. And that's sort of the definition of precision medicine. Different people are different. Their genetic makeup is different. Why should we have one drug treat everybody, you know? It just makes sense if you think about it. And this has actually been done pretty well for cancer, but it's never been done for GI people with GI problems. And so that's what makes our company unique and really exciting. Another aspect of my company is we develop blood tests, so diagnostics. So people that have get gastrointestinal disorders, they go to their doctor and they want to know, is my drug working? How's my health? Do I need to change medication? We take blood, bring it back to our lab, run it on a bunch of different experiments to try to understand what's going on with them. And then we give the data back to the doctor, the actual, the physician, and then they can make decisions on these patients and how to do them. So my company actually has two big buckets. One is blood testing for doctors, and then the other is developing a new drug. And this is really what I'm doing, is developing a new drug. And we're really close. We have an antibody that we're going to put into the clinic this year. So this is a big deal because we're all, all of biotech and drug discovery is trying to get your drug into the clinic, actually testing in humans, because there's a lot of work that goes before actually testing in humans. And so we're really excited. That should happen this year. We're going to have what's called a first in human trial, where actually healthy people volunteer to take these new drugs to help people. They take them, just be sure they're not going to get dizzy or they're not going to start throwing up. And then once the healthy people take them, then we start getting people that are sick with the disease and see if it actually works. So on my company, I'm really excited. We're working really hard because we have to have all sorts of documents and data and just giant stack of papers that somebody has to review at the FDA. So me and my team, we're working really hard to get all that stuff together so that we can go to what's called the FDA, and they can say, oh, you're good, let's try it, testing it in humans. Or they go, you need a little bit more data before we try doing it in humans. What you don't want is them to go, so don't worry, that's not going to happen.
Evan had a good question that's related to what you were talking about. He was wondering how long your company has been working on the drug you're currently working on.
Oh, that's a good question.
Yeah.
So normally it can take up to 10 years to develop a drug. We have done it pretty fast. So the company has actually been around. It changed names, but as the company, when I joined, it's been around for maybe 3 years. But before the company, this is a spin-off from actually an academic lab. So a lot of, not all, but some companies start out in an academic lab. What is an academic lab? That's like a university or a hospital, not a company, but these guys are working really hard to understand the biology of a disease. They had this idea for this drug, and so they started the process. And they were so excited about it and said, hey, let's take this drug and start a company. And so that's what our company is a spin-off from somebody at actually a hospital in LA called Cedars-Sinai. It's a very famous hospital. Since it's actually in Beverly Hills, so a lot of TV stars go there. But so it's a spin-off from that. So it's probably maybe 6 years or so to get to this point. But as far as the company, it was maybe 2 to 3 years. And the kind of drug that we're developing is what's called a therapeutic monoclonal antibody. So it's different than other kinds of drugs, which are pills that you take. This is actually a drug that you typically have an injection for it, but it's very different. It's actually a protein. It's a big antibody protein, whereas a lot of the drugs like Tylenol and stuff are really teeny tiny small molecules. And so the development time and the process for each are very different. My background is actually the little small molecules. And so this is really cool. I'm starting to learn more about antibodies and how they're made. And actually these guys are you can go really fast with these guys. It's amazing how fast you can actually develop an antibody and go into a clinical trial. So, it's been fun. I really enjoy the company. It's changed. We used to be really small, like 20 people. And there was like maybe 3 or 4 of us in the lab when it first started. So we're really small. And then all of a sudden, huge, huge, like 200 people, but real much bigger. So it's been fun to be a part of it from the very beginning when it was a small company to now growing bigger and bigger. And eventually, when we get our drug approved, we'll be huge. So we're looking forward to that. Really fun.
Yeah. Cameron was wondering what is an assay?
An assay, that's a very good question. When you're in drug discovery, all about assays. So assays, you can kind of equate assay as an experiment. Assay is a set of conditions where you're looking to understand a certain process. So for one of the assays that we do is a cell, and we treat a drug, and we want to know how well that cell is responding to the drug. Is this a good drug in that cell? Is it doing what we want to do in that cell? And so what we'll do is we'll have the cells, we'll have the drug, and then we have what's called a readout. So the cells will either change color, or something will happen to them that then we can read and assign a number to. It's called quantify the effect. So doing an assay is understanding a process and then getting a number from that so that you can say, okay, this drug is better than that drug because this number is higher and this number is lower. So an assay is a way to quantify a question. And assays come all different forms. You can do them in cells, you can do them in proteins, we do them in blood. So human blood, they donate blood, and we look for different assays in blood. So assays, you can think of assays as sort of like an experiment, a little mini experiment. And we do actually thousands of assays. In the next portion, I'll talk a little bit about what a lab looks like and what we actually do when we do assays.
There's one other question that was correlated to this section. Landon was wondering, why is it so hard for scientists and take so long for them to find a cure or find a solution? Why does it take so long to do the process?
The real reason is life is so complicated. I mean, I look like a normal person, right? But if you look at what's going on in each one of my cells, in my organs, in my eyes, and my brain, it's so complicated. We're not even close to understanding what's actually happening with life. I mean, we know a lot of things. We know about cells, we know about DNA, we know about this, we know about that, but really there's such a large amount of information, so complex that it's really difficult to develop drugs and totally get something that works. The other thing is that people are different. Like I said, there's genetics. Genetics of different people are different. What drug might work for this person may not work for another person. So, one of the cool things about science is there's so many unanswered questions. Nobody should ever be like, oh yeah, we figured that out. We figured out how the heart works, no problem. No, it's so complicated. That's what I think makes it so interesting. There's always questions to ask. There's always things to discover. But yeah, it's super complicated. Yeah, it's really interesting. The more you learn about biology and science, the more you realize, oh man, we do not know everything. So when you're, like when I was a kid or something, I'd be like, oh yeah, DNA, we got that figured out. But then I started taking classes like, oh, that's pretty complicated. Oh, you mean there's regions of the DNA that don't actually know what they do? It doesn't look like the spiral. It's actually, the more you learn about science and the more you get detailed in it and higher you get in your education, you realize, oh my gosh, we do not know everything about science. There's so much more to learn.
Well, this kind of starts off the next section of the questions. Madison was wondering what your favorite part is of your job.
Okay, my favorite part of my job is what's called the discovery process, finding something new that nobody's ever done before. And for me, currently, it's finding a drug that works that nobody's ever done before. So I'll go into the next section. The next section is going to talk a little bit about my background, how I got to be where I am, and give you a better sense of, as a kid, how did I get here? And I think that kind of feeds into this whole idea of what I like about science. So let's see here. We'll start with this. Can you guys see that? Can you see my screen?
I can see your screen. How drugs are made screen.
Okay, we're done with that section. Okay. So I'll start by talking about a lab and what it's like every day in a lab. And I'll tell you about how I got there. But one thing I do want to make clear is that when you see pictures like this, okay, there's these two people, they look like models, like they could be in a Hollywood movie. They've got their stuff on, there's DNA floating around the lab. They're working on with these, is this what a lab looks like? This is not what a lab looks like, okay? What about this? you know, they have their little test tubes, they're doing this, they're doing that. There's interesting things. That's not what a lab looks like. Labs are actually really big, and messy, and complicated, but there's a lot of cool technology. This is not what a lab looks like. This is where we get into the idea of assays. So if you look on the left, this is what we think labs look like, right? Test tubes, a little glass thing, we do something there, and then it changes color, and aha, we found a drug. This is what, you know, people used to use. Now assays in these things, these are called microplates. And so this one on the left has 96 wells. So really quick, what's 8 times 12? 96. So it's 8 this way and 12 that way. Now, if we take that and we multiply 4 of these in the same, you know, footprint, you get this thing in the middle. That's a 384 well plate. So 384 individual assays to do at the same time. Now, the last company I worked with took it a step further. They took 4 of these 96 well plates and crammed them together so we get this one over here. Do you see that? That's 1,536 individual assays that you can do in this little plate at the same time. It's pretty cool and complex. Now, can you imagine these guys trying to work in one of these with those big funny pipettes and stuff? That's not going to happen. What you actually need is automation. So I wanted to show a little video just to give you guys an idea what that's like. It's pretty cool. Ruby. So at my last company, we used a lot of automation, and we did a lot of our assays in those little teeny tiny plates. I froze. There you go. Can you guys see that?
Yes.
Yes. So this is really cool. This is an automated system where you set it up, walk away, and it does assays in those little teeny tiny wells, 1,500 at one time. And that's just one plate. And then we have hundreds of plates like that that we test, you know, over the course of a week. So we're able to 1 million assays in less than a week. So 1 million chances to find a new drug is what's going on here. Each one of the wells could be the next drug. We don't know. And that's really what I love about discovery, drug discovery in particular, is you never know what you're going to find. We may find the next cure for IBD or Crohn's disease, or in this company, we were working on neuroscience, so trying to make the brain work better. And that's what you're seeing right now is an actual high throughput screen to try to find the next drug that will make your brain work better. So my lab currently that I work in right now is in, sorry, let's stop sharing. So the lab I work in right now at Prometheus is in the Miramar area. So it's really interesting. Biotechs have what are called hotbeds. So this is different regions of the US. There's a whole bunch of biotechs clustered together. And one, of course, is here in San Diego. So San Diego has a whole bunch of biotechs right around La Jolla. There's also a whole bunch in San Francisco. And then there's a whole bunch in Boston. Those are what are called the 3 hotbeds of biotechs. There's hundreds of them in those little regions. And it's important. The reason why they're so close together, anybody know why they might be so close together and not just spread over the whole country? There's 2 reasons. Number one is they tend to be really close to highly academic places that have really top-notch academic universities. So you have in Boston, you have Harvard, you have MIT, you have all sorts of stuff. In San Francisco, you have, you know, UC San Francisco, you have Stanford, you have Berkeley. San Diego, we have our own little cluster of really top-notch schools. And one of the ones that I went to is called UC San Diego, University of California, San Diego. Go UCSD, go Triton. So that's reason number one. Number 2 is biotechs work together. It's a team sport. We have to be able to talk to each other. And so there's a whole bunch of biotechs close together and not spread out over the entire country because we need to talk to each other. We need to visit other labs. We need to, take our DNA and go find Mrs. Bourne and say, I got this DNA, I need it sequenced. Can you get Elim Bio to sequence this for me? So you need people close together to do that. So there's other little regions around the country that are biotech clusters, but those are the three. And I'm lucky enough that there's one in San Diego because, San Francisco is really crowded and Boston is cold. So San Diego is the best place for me.
I agree. We're very lucky to live in San Diego. So we had a poll that went up, Dr. Rojas, and I don't know if you can see it, but it was what kind of technology? And,
yeah, like I said, we use artificial intelligence. There's just because, if you're interested in computers, you can do a lot of things with computers, right? I mean, you can work at Microsoft or Google, but you can also work at Prometheus Biosciences. We have this one guy, he's a genius. He does artificial intelligence, machine learning, and data analysis to understand which SNPs we should go after. I mean, he's a computer guy and he works at a company like mine. So there's a place for, each one of these different STEM, we talk about science, technology, engineering, and math in different companies. And so if you're thinking, I'm a math person, I really like math, but that just means I'm going to be stuck teaching math. That's not true at all. You can really branch out and go to different types of companies if that's what you want to do.
Awesome. Great. Let's see if I have another question. Oh, I have a question that came across about the FDA, and maybe it's kind of correlated right now because people are thinking about new drugs are being created. Has the FDA ever denied a drug that was created, and why would they do so?
Oh, I have an interesting story for that. My first job after I got my PhD was at a company. And when I first joined the company, they said, we have a drug that's right about to get approved. So this is really going to be good for us. The FDA is going to tell us on this date that it's a go. And so I was like, great, I'm going to join this company. They're going to finally have this drug approved. It was for insomnia, for sleep. So I'm there a few weeks. This is my first industry job after I get my PhD. I'm really loving it. And guess what the FDA said? The drug was not approved. What they said is, we could probably approve the drug, but you need to do X, Y, and Z. And unfortunately, X, Y, and Z meant millions and millions of dollars, and there was no way that we could have done it. So that actually happens a lot. A lot of drugs do not get FDA approval. And that's because, like I said, biology is so complex. Sometimes we think it's going to work, and finally, when we test it in people that are sick, it just doesn't work. Unfortunately, that's the case. Actually, I hate to say this, but most drugs that go to the clinic do not get approved, unfortunately. So that's my story for not getting approved drug.
Have you had drugs that have been approved?
No. In fact, this is actually very common too. If I've worked with a lot of scientists over my years, 12 plus years, I guess, I've been in biotech for now. I've met a lot of scientists, and actually not that many have ever worked, really closely on a project and gotten FDA approval. I mean, there are some, but majority of people are like, man, we tried, we couldn't get it to work. We tried, couldn't get it to work. So failure, unfortunately, you have to, there's a large failure rate, and that's fine. You take it and say, I'm gonna continue, and you can't let that bring you down. You say, I really want to find a drug. I really want to help these people, so we're going to continue. And I'm not going to get into the politics of drug pricing, but drugs are expensive. Some of them are too expensive, but they are expensive, and the reason is people are like, why are they so expensive? Because we fail all the time. It costs money. I mean, we try this, it fails. Well, that costs a lot of money to fail. So when we do get it to work, it actually kind of has to pay for all the failures too. Just because it takes so much time and resources to make a drug, people don't realize the time and resources to actually get a drug approved and make it. And so if they did, they'd be like, oh, wow, no wonder they're so expensive.
Yeah. Has there ever been drugs that the FDA approved and then they thought later, oh, That was a mistake. We shouldn't have approved that one.
Yeah, that happens, unfortunately. And it happens more like, you know, years back than now, but sometimes that happens. Not very often, because the FDA is very careful with what they approve. It really has to be, I mean, the most important thing is that it's safe and that it works. But out of those two, it has to be safe. That's what clinical trials are all about. Is it safe? And does it work? Because they're not going to, if it's safe, but it doesn't do anything, they're not going to approve you, right?
That's great. Well, we're kind of moving on to the next section about how you became interested in science and your background. But I'm going to throw up just a funny poll to see what people think about what you were like as a kid before you did.
Oh, yeah.
And what you did all the time when you were a kid. So there's another poll. And just take a guess. You're not going to know the answer to this, friends, but just take a guess of what you think that he liked to do when he was younger, about your age.
So I answer the question first, and then I'll tell you how to be where I am.
All right.
I don't want to give anything away.
Okay, we'll give it one more minute. While that's happening, how did you become interested in science?
That's a good question. I'm going to show some slides that kind of talk a little bit about this. Let's see if, can you see that?
So far we see the microbial plates. Looks like most people voted. I'm going to go ahead and.
I must have a lot of people that know me watching this webinar, because that's absolutely right. I used to skateboard and listen to punk rock music when I was a kid.
You mean you used to sing all the time?
I'll talk a little bit about that. The path to how I got to be a scientist, and it's not as straightforward as you think. You know, you're like, oh, scientists are geniuses. They're always smart. They always did good in school. You know, there was a clear path. They were valedictorian. They always got A's. It's not true at all, and I'll talk a little bit about that now. So I started way back. You know, I was at Laverne Heights Elementary, where I grew up in Laverne. Laverne's the city outside of Los Angeles. It's still in Southern California, but it's outside Los Angeles. After that, I went to Ramona Middle School. After that, I went to Bonita High School, all in Laverne. And then, so if we think about these three schools that I went to, a lot of the kids, you know, that are on the listening right now are either in, you know, elementary, middle, or high school. And I'll tell you guys a little secret. Don't tell anybody that I work with, but I was a terrible student. I was not a good student. I did not get straight A's. I didn't even get straight B's. I mean, I was not a good student. And there's a few reasons for that. One is I would much rather be skateboarding and listening to punk rock music than studying when I was a kid. But the other thing is I just couldn't find something I was really interested in and engaged in. But you know, during these early years, I was always interested in the way things work. And there was a book that I had. It was called The Way Things Work, and they still make them. And I was really interested in that book, because what it did is it took things and took them apart and told you what's going on in them to do what they do. And that's one of the things I used to like to do as a kid, was actually take things apart, try to figure out how they work. And I was never so good at the putting back together part, but I like to take them apart and try to figure out how they work. So I did that, and I was interested in biology. One thing I'll say is my dad is actually a doctor. He's a medical doctor. And so while I was growing up, I would be interested in what he did. You know, what exactly are you doing? You know, what's science all about? So he really exposed me a lot to science at a young age. But the other thing I want to say is, since I didn't get straight A's and I didn't do that well in school, I always didn't think I was that smart. I never thought I was, actually, I still don't think I'm that smart, but growing up, I never thought I was really smart. I wasn't in any of the gifted classes. I was, when I took standardized tests, I did okay. But I'd never let that slow me down. After I graduated from high school, that's when I really put in the extra effort and say, oh, now I have to actually do something to be successful. So that's when I started to turn things around. The first college I went to was actually a junior college, Mesa College. So this is a 2-year college. It's kind of like Palomar College that's here in San Marcos. You can go there for 2 years to get you ready for a 4-year college. And so I did that for 2 years, and then I went to UCSD, a 4-year college. And it was actually a really difficult transition for me going from high school to college. The reason was I never learned how to learn in high school. Does that make sense? You actually have to learn how to learn. Okay, I need to read, take notes, do this, do that, be rigorous. I didn't really do that so much in high school. So I had to figure that out in junior college. And some people learn that faster than others. It just took me much longer to learn that. And some kids, I volunteer at the elementary, Richland Elementary, go Mustangs in the city. And there's some kids that are really, really bright. And I'm like, man, you guys are going to have if you want to be a scientist, it'll be easy for you. And, but it wasn't easy for me. And it doesn't have to be easy. I mean, I'm still really successful, but it just, took me a little bit longer to learn things. So after Mesa College, I then went to UCSD, and oh my gosh, UCSD was so hard. There's really a lot of smart people there. You talk about, people like valedictorians, which are, the top of their class in high school, people that are in gifted classes, they all went to UCSD. And so I was taking classes with these guys, and I was, one thing I noticed is, I had to study like twice as much in order to get an A on my test. Or some people it was really easy. For me, it wasn't so easy. But who cares? I mean, we all learn at different speeds. And so, at that point, I was like, I really like the science. I really want to, turn this into a career. And actually learned that in Mesa College. So I actually went to college thinking, I kind of like the mind and how the mind works. Maybe I want to study psychology. And when I started taking psychology class, not to, make fun of psychologists or whatever, but it was a lot of talking, and some people would say something and they're like, yeah, that makes sense. And then somebody would say the complete opposite and like, yeah, that makes sense. And I was like, oh, I need this to be a science. I actually want to know the answer. I want to quantify things and really understand the answer. And so that's why I actually switched from psychology to neuroscience. Neuroscience is much more rigorous and what the brain is working in the cells and stuff. And so I was at UCSD, I studied neuroscience and animal physiology, which is how the body works. So I graduated there, and then actually worked for 2 or 3 years at UCSD before I finally said, what, I like working in the lab and it's fun, but I kind of want to be my own boss. I want people to listen to me instead of people telling me what to do. I'm like, I could probably do this. I think I can run the show here. So I said, I'm going to go back to school and get a PhD. And so I went all the way across the country to North Carolina, go Tar Heels. So I went all the way across the country to UNC Chapel Hill to get my PhD. One question that I get asked a lot is, what did you do in graduate school? I was in graduate school for about 5 years. Some people take longer. Some people are in graduate school for 6 or 7 years. What the heck was I doing that whole time? And so this slide, in a nutshell, says what I did. So what did you do in graduate school? Well, graduate school is all about finding a question and trying to answer it. It doesn't have to be a profound question. It's not going to be like, how does the brain work? Or is there a consciousness? It doesn't have to be a profound question. It could be just a little nugget of a question to get us further to these profound questions. And so if you look at this little cartoon I have here, this talks a little bit about what my actual research was about. This is a cartoon of a cell. This is the outer cell membrane, this little bubbles. It's a bilipid layer. And I made this. It's another fun thing about grad school is you make a lot of cartoons, which are, I enjoyed making. So this is a cell. This right here is what's called the G protein coupled receptor. And then this right here is a ligand for that receptor. So this thing binds, it activates it. And what was known in the literature is that there's two pathways by which these guys can signal downstream to these what are called Rho proteins, Rho GTPases. They can either go this way, the blue one, 1213, can engage this family of proteins, and then that's how they activate it. But what they also found is they can go this way with this protein, and this protein usually activates this, but they found that this protein can also activate that. But they didn't know how it activated it. See the question mark? Graduate school is all about finding these question marks, as little as they may be, and trying to answer them. So that's what the question mark that I tried to answer. And so on the next slide, you know, a lot of my work can be distilled down into this figure. This figure, and you make figures like this in graduate school and science and stuff. And what this figure does is this is the protein down here. If you start at the left, that's what's called the N-terminus. And on the right is the C-terminus. And then these squiggly lines tell you for each of those amino acids, how important is it? Are they conserved in nature? How important are they? And so you can see over here on the protein, it's not so important, right? And then you get to what's called the DH domain, where we know this is important, then it's really big and red. And then you get to the pH domain, part of it is pretty important. But if you look over here, do you see this? That little piece is important. What's going on after the pH domain? It's really important. The reason that we think it's important is because it's conserved. So frogs have it, fish have it, mice have it, rats have it, chimps have it. All these species have the very similar sequence here, but we don't know why. And so what my PhD was to figure out that little piece is the part of the puzzle here that connects that to that. So I won't go through this, but basically if you have that little piece there, you get a big red bar, you take that little piece off and you look, that bar goes away. It's no longer active. Graduate school is fun. One of the cool things about graduate school is meeting people and actually having a mentor. So when you're in graduate school and you get a PhD, you work in someone's lab, you know, a big professor. And the lab that I chose to work in is this guy named John Sondek. So Dr. Sondek was a lot of fun to work with. As you can see, he likes to mess around. He likes to joke around. So it was really fun working with him. And I learned a lot about the rigor of science and what it takes to be a good scientist from people like him.
Great. One of the questions was, what was your favorite subject when you were in school?
So when I was in high school, I liked biology. I really did like biology. I didn't know I was going to make a career out of it, but I did like biology. I thought it was interesting. I really was kind of fascinated by, like, you know, what makes something alive? Why are we alive and like a rock is not alive? Like, what's the difference? What's going on there? And so biology is the study of life. And so I really thought, wow, I mean, how fundamental is it to study life? And so I was really interested in biology.
I like biology too. I put up a silly poll for people to guess what your favorite food is. So we'll see what people guess. It's just something silly, because I'm sure, do you have any pets or anything that you wanted to tell people about?
Yes, I have some pets. In fact, I have 2 dogs. If you guys have a chance, I'll show you my 2 dogs.
Yeah, I think we have time, and then we'll be wrapping it up soon, but that would be fun.
Here's one dog, and here's my other dog.
Oh, they're so cute.
Here is Crosby, and over here is Elvis. And yeah, I have 2 dogs. And you know, when I'm not working in the lab and studying science and stuff, you know, I'm just a regular dad. I have 2 kids. I have dogs. And one thing I did want to mention is actually, usually I don't like to tell people that I'm a scientist. And it's kind of dumb, but this goes back to the whole genius smart thing. I don't think I'm a genius. I don't think I'm like really, really smart. And and a lot of people have this stereotype that if you're a scientist, you're a doctor, and you have a PhD, you must be brilliant at everything. And the truth of the matter is I'm just a really kind of ordinary guy that yeah, I worked really hard and spent a lot of time to get where I am. But, you know, back in elementary or middle school, I was just a normal kid. And so I don't always like to tell people I'm a scientist, because then they'd say, oh, well, I must be a genius, or, you know, the next Einstein. And that's not the case. There are, you know, a lot of scientists are really smart, but you don't, you know, I don't like people to assume that I'm smart. So it's just, you know, because there's a little bit of stereotypes, you know, oh, he's a scientist, so he's a genius. And, you know, I don't like to play into those stereotypes. So sometimes I don't tell people that. Eventually it comes out. Oh gosh, I have to, they're going to ask me all these questions about this and that, like COVID, you know.
Yeah, I'm sure there's people who have questions about COVID.
So bring it on. You know, a lot of people like to ask me about that because I'm a scientist. So yeah, I understand biology, but I'm not a virologist. So if there are questions about it, I'd be happy to answer them.
Well, we did have one question about COVID-19. Landon was wondering what the symptoms are, if you know more about that.
That's a good question. So I will say, you know, leading into this COVID-19, and, you know, one of the reasons why we're having this WebEx in the first place is because we're trying to do something. We're trying to do what's called flatten the curve. Mm-hmm. So by flattening the curve, what does that mean? And that's something really important. I'll get to the symptoms in a second, but I do want to state something here that over time, so, you know, you're thinking about time, people are getting sick. People are getting sick. Oh, a whole bunch of people are getting sick. When there's too many people sick, there's not enough place in our hospitals to help them all at once, right? There's just too many people in the hospital to help them. And so by staying home, what we're doing right now, you know, I'm staying home. I don't go to work. I work from home now. I actually don't go in the lab anymore. I have teleconferences and phone calls from home. And so by staying home, not everyone's going to get sick at the same time. We're going to spread it out so that they can deal with it in the hospitals. So the symptoms of COVID-19 are very common, are very similar to things like a common cold or the flu. You'll get a fever, you'll cough, and have trouble breathing. There are two additional new symptoms that I thought were kind of interesting. One of them is you may lose your sense of smell and taste. So you may not be able to smell so great, or food may not taste the same. That's one symptom. And the other symptom is like GI problems, like your stomach kind of hurts and that kind of thing. The primary symptoms that everyone gets is a fever, cough, and a tightness of the chest. And don't be scared. I want everyone to understand that sounds like things are scary. This is not the end of the world. People are not going to turn into zombies. This is, you know, we're all staying at home to help, you know, slow the spread, but don't be scared. We're going to get through this fine. And as long as you distance yourself from other people, don't go out and play with all your friends that you miss. You're just going to have to wait for that. We can slow the spread, and we'll get back to normal really soon.
One of the people listening wanted to know if your company is trying to do anything for COVID-19.
That's a good question. So we don't do, you know, the drugs that we work on don't really help, you know, viruses. So it'd be really, we're not in a position to make a drug for people that have COVID-19. But one thing that we can do is some of our blood tests. What we're doing is we're actually letting patients that can't afford them right now, they can have it for free, because they need to know how well they're doing. They can have the test for free. We're like, you know, a lot of people are losing their jobs. Just take this test for free. We'll pay for it. The other thing is a lot of people are not able to go to the doctor to have, you know, certain exams and stuff. And so what we do is we have a blood test that they can take instead of having to go to the doctor and do all this stuff. We actually pay for somebody to go to their house and then take a little blood, and we'll look at it to see how they're doing. So we are trying to help people with COVID-19. Sure, we're not making a drug for them, but for our patients that our company focuses on, people with GI problems, we're definitely helping them as much as we can.
That's awesome. There's one more question, and then I think we're gonna be wrapping it up soon. Well, we'll make sure to share your favorite food poll results. But there's one more COVID-19 question, and it was, why are younger people handling it better than older people?
That's it. You know, this is one of the things about science. We don't know everything about everything. So we don't know. I don't know. The cool thing about science is you start with a hypothesis. You say, okay, maybe it has something to do so that the virus gets in and attacks your lungs and your airways. Maybe there's something about young people that their cells in their lungs are a little bit different. And so what you do is, that's your hypothesis. You make a test to see if you can answer that. But there's a lot that we still don't know about this disease, unfortunately.
Yeah, this is new. So here's the share results about what people thought you might like to eat.
Yeah, that's pretty close. Yep.
Pretty close.
Kale is at the bottom of the list. I actually like a lot of those things. I should say, you know, my favorite is probably California burritos and In-N-Out burgers, followed by things like cookies and sushi. And, you know, carrots and hummus I actually do like quite a bit, but In-N-Out burgers are definitely my favorite. And so it looks like, man, a lot of people here know me. It's not fair.
Maybe it's their favorite too.
Yeah.
So I'm going to go ahead and make sure that we talk about a little bit about next week. And I wanted to thank you before I do that, Dr. Rojas. It was really great talking to you.
Thank you. Thanks for joining. Stay safe.
Thank you very much. And I wanted to let everyone know that next week we also have another webinar that we're going to be hosting, and it's with Dr. Sushmita Kumar. And Dr. Kumar has a really famous location where she's doing research, and she is working on biofuels, and her research, she'll tell us more about why her research is like microscopic Pac-Man. So that would be kind of fun to hear about. And then we're going to have the call on Thursday next week, and it'll be at 1 o'clock on Thursday. And I'll put up a link in an email so you can see how to register for that. So everyone who was here, I have your email, and I'll send you a link so you can register for that class too. And it's still free. So if you want to find out more about how microscopic Pac-Man works next week. And thank you very much, Dr. Rojas. We really appreciated you and your time and all the effort you put into being here and being here for our students. And we really appreciate your time.
Yeah, and thank you, Mrs. Bourne, and thank you, Elim Biopharm, of course, for sponsoring this event.
Of course, of course. Well, have a great day, guys. Stay kind, stay curious, and wash your hands.